All right, in this lesson, we're talking about other types of inheritance that aren't Mendelian. All right, starting with codominance, and it is different to Mendelian inheritance, but has the same a few elements that are very similar. They have both alleles that are different variants of the same gene, but they're going to be expressed equally and independently of each other. So, for example, we might have an allele for a red color, we might have an allele for a blue color, and when they are both there in the heterozygous combination there, they are both seen. So some examples in real life. We have cows, so brown cows and white cows become brown, white, spotty cows. Same thing in chickens. We've got white chickens, black chickens, and they become black and white speckled chickens. And it's pretty common in nature as well to see. So you might see leaves that are yellow and green as a mixture, and quite often flowers are this speckled situation as well. All right. Blood grouping is the most common version of codominance, a really good example of codominance. And what happens on the red blood cells or the erythrocytes? So we have antigens. So group A have one type of antigen around the outside of their cell surface. Group B have a different type of cell antigen on the outside of the cell surface. And when you are group AB, you actually express both equally. If you are group O, you do not have any antigen there at all. Right, so we talk about donors and receiving when we donate blood, and it's all to do with these types of antigens. And there are other blood groups that have different antigens as well. So if we are referring to this particular gene of expressing an antigen as a capital I, then this is one variant, the A variant of the A antigen there. So you can be a homozygous dominant here. It's not really dominant anymore, but here it does express its dominance over this allele here that would express no antigen. So we'd both have a phenotype of A here. Same thing happens with a B. We can be homozygous for the B antigen or heterozygous with the recessive antigen here. But once we start expressing both of those dominant antigens, we end up getting both of them expressed equally. And if we have that recessive one there, we don't get any antigens and we have the group O. So it is similar to the dominant recessive in that we still have one that dominates and one that kind of shies away. But what happens if we get that combination is that they are both there. So that's the main difference. So for example, my blood type is A. I know my dad is an O and my mum is an A. So really, what is my genotype? There isn't really too much of a combination available there. If my dad's an O, he's definitely got the recessive gene, so he must have two O alleles or those little I's that we saw before. And my mum is a phenotype A, so she's got two options there. She can be homozygous for the capital I with the A antigen, or she can have the capital I with the A and the small recessive one there. So if we talked about mixing blood types to create offspring, then these are our possibilities. My dad would be here. They're the only things he has to offer to the gametes, and there's one combination, and it would only result in me being AO, so heterozygous. The only other option there is if my mum was started off as heterozygous and my dad was obviously that homozygous recessive, then 50% of the potential offspring would be O. Right, so I'm probably one of these. Right, remembering the phenotypes of offspring can be different from their parents. For example, if we're going to cross some people who are heterozygous, there's an A and there's a phenotypic B person, we're actually going to end up with all four blood types here. We've got an O, right, from mixing these two. We've got an A phenotype, a B phenotype, and an AB phenotype. So 4 potential outcomes and 4 very different outcomes. Same thing over here. If we're going to cross that recessive one with the codominant situation there, what you're doing is going to see 2 potential options there of A's and B's, and they are both different from their parents. All right, an incomplete dominance, different type again. We're looking at heterozygous alleles, but are expressed as a hybrid phenotype. So we're talking different to codominance, where in codominance you see both of the phenotypes. Now we're actually going to combine them and get something that's different from the parent. So for example, here in codominance, if we mix the blue person with the yellow person, we're going to get a mix of both. And the difference here is that that combined hybrid phenotype is going to be coming out in incomplete dominance. Very, very common in nature, particularly in flowers. So if we can compare our codominant here to our incomplete dominance using the same types of parents, we're just handing down the same types of alleles, but we're expressing them in different ways phenotypically. Right, multiple alleles are also called polygenes, They are phenotypes determined by more than one gene. We like to simplify so we can explain inheritance to you that say, okay, well, here's this tiny little gene and it determines what your hair color is. But in actual fact, stuff is very, very complicated. And because we have multiple genes that are potentially lending itself to one phenotype, we can't really talk about it that easily all the time. And we get a continuous variation in a population. So if we just talked about one gene, we get those 4 outcomes. It's like a simple monohybrid cross in a Punnett square. If you start talking about 2, you know that the gametes that are produced, suddenly we've got a lot more. If we start talking about one phenotypic thing that is expressed because of 3 different genes or a combination of those 3 genes, then we're going to get more and more and more potential gametes and therefore more and more and more potential outcomes when we cross individuals. And what that leads to is this huge spectrum of variation, because that's really what all this stuff is all about. It's about variation. So you can see immediately here, skin color is not just going to be controlled by one particular gene and we've got this outcome or that outcome. We're going to get so many different outcomes because it's controlled by a number of genes on a number of different chromosomes. Same thing here with eye color. Eye color is incredibly complex and it's not just one gene on one chromosome. All right, sex-linked genes are very, very interesting because as between those 2 different biological genders there, we're going to see huge differences because of what our sex chromosomes are. Females are the homogametic gender. Homo meaning the same there because we have our sex chromosomes are exactly the same. We've got an X and we've got another X. Obviously, contrasting that to males, you are the heterogametic gender. So therefore, you've got the 1X and the Y. The heteromere, they're meaning they're different. So even though they're homologous pairs, right, they're not going to be exactly the same. So the genes on the X are not going to be the same as the genes on the Y. And what happens then is that if there is a defective gene on an X, for example, in a female, the other one can say, right, that's fine, I'll take over. If there's a problem and there's a defective gene on the X in males, unfortunately, there's nothing else there as a backup to use. So the recessive alleles on an X chromosome must be expressed in the males. There's no way it can push that one under the rug and use the other one because it just doesn't exist. So in this situation, what this means is they would inactivate this X and the genes would be taken from this. So this is our active X and this is our inactive X. Unfortunately, with only one X available, has to be the active X. What this means is that if there is a real problem, so in this case, the defective allele, it will absolutely be expressed in a male who inherits it. So obviously the father is passing along the Y chromosome in its gametes. In this particular case, it's the only way that we can get a male offspring. In this particular case, obviously the male is donating that X there, but the mother is always donating an X, okay? That's all she's got to give. It's whether or not she's going to donate that defective allele. In this case, she has, but we're going to inactivate that and make this one the one expressed, and this becomes known as a carrier situation. The disease or the disorder is not expressed phenotypically, but it does sit there and it does exist. It's just kind of quiet and silenced at the moment. Unfortunately, if the male gets it, it can't be quiet and silenced. It's the only possibility for expression. So colour blindness is one of those situations that is expressed on the X, and therefore it is more likely and more common to find males who are colourblind. Females can be colourblind, but they've got to be pretty unlucky to be getting a defective gene from both parents, that the inactive X has the defective gene and the active X has the defective gene. So the things that are not meant to be there, so the rods and cones in your eyes that are meant to see the colours there, are not being made. Another one is haemophilia, and haemophilia is very common, but mostly in males. You very rarely see it in females, and that's the same reason. So if you are a male, you are going to pick up that affected phenotype because you've picked it up, the defective X gene, and mothers are therefore the carriers. Okay, so if it becomes very prevalent in a family, it's because the women there are carrying it and it's a silenced situation, and then most of the males that are going to pick up that X from the parent there, we're going to lead to some problems, okay?